Uh, a very good afternoon, everyone. As always, this is an absolute pleasure giving a talk in an ISEB event. Usually my slide starts off with a photograph of mating fruit flies, but this is the first time there is a mating beetle. At least they are mating, so my interest has not changed. I haven't left. That's still my bread and butter. Yeah. So these are zygogramma beetles, and just to give you an idea, this is the very hardworking and talented student, Roby. Whatever I'm going to present, this is all his work. Just a little bit of information to start with. These are specialized herbivores on Parthenium. It is non-native to India, but was introduced in India to control Parthenium. Now you can find these beetles everywhere. They have a pretty mundane lifestyle. They're promiscuous and there is little sexual dimorphism, but they have a very interesting mating behavior, and I'll come to that in a little bit. Right, so Roby collected a large number of these beetles, brought them to the lab, and created a stock which was maintained in a very tightly regulated ecology. I'm not going to walk you through this, but if you're interested, I can talk about this later on. All the experiments were performed using beetles generated from these stocks. Yeah. So the interesting thing about their mating behavior is that they have an inordinately long mating. So normally in insects, mating ranges from a few seconds to several minutes, and in some insects, for example, these ones listed here, they have mating ranging up to 6 hours, 10 hours, even 24 hours. And our beetles, the Parthenium beetles, they often mate for more than 6-7 hours. So that is where our interest lies. And Roby started asking the question why they mate for such a lengthy time. Right. So first thing that we took up is we started looking at mating behavior, actual mating very closely, and it turns out that the mating in these beetles is actually not one long mating, but it's a series of repeated intromation punctuated by these intervening short periods where there are some interesting behavior, something called leg rubbing and leg biting and so on. I'll come to that in a little bit. But very importantly, just to rule out the possibility that since we are observing these behavior in the lab, these are not lab artifact, we actually repeated the same observation in lab as well as in semi-natural condition, and both showed the same pattern that mating is pretty long, often ranging beyond 6-7 hours, and each of these long mating is actually a series of, on an average, 50 to 60 intromissions. Right, so the question is, why do they need to mate long, and why such weird behavior of repeated intromission? And this is the two questions, these are the two questions we started off with. If you look at the literature, it's almost like a foregone conclusion that if you have an insect, or any animal for that matter, mating for a very long duration, it has to do something about mate guarding, copulatory, post-copulatory, whatever it is. But there are a whole lot of other adaptive theories that should be non-mutually exclusive and usually are not tested, and that is what we started off with. We tested all different possibilities. Just to name a few, sperm transfer can be one limiting factor, transfer of accessory gland proteins, seminal fluids, and even nutrition. So these are all different possibilities that we started exploring. To investigate some of these theories, some of these predictions, Roby used a large number of interrupted mating experiments. The idea is very simple. You start a mating pair by introducing a male and a female together in a mating arena, and then wait for a certain number of intromissions to reach. For example, you can see here, I1 is immediately after one intromission, I15 is after 15 intromissions, and so on and so forth. Interrupt the mating by physically disturbing them. And then here, the first thing that Roby looked at is the number of sperm transfer and the number of eggs produced, hatchability, and so on. So this is looking at sperm transfer and fertility. And it turns out that single intromation is definitely not sufficient to reach the maximum capacity. At least 15 intromissions are required. When actually this saturation is reached is not very sure, not very clear, because we don't have these intervening treatment levels. But beyond 15, the more number of intromissions does not make any difference in terms of sperm transfer. The same thing also applies for fertility traits. Fecundity and hatchability does not improve beyond 15 intromissions. Right. Now, if these repeated intromission, long mating has something to do with nutrient transfer, then the simple prediction is that an unmated female should live shorter under starvation compared to a mated female. And similarly, if you interrupt mating at different intromission numbers, then those should also scale accordingly. And that is where we ran these experiments, looking at 4 different levels: unmated, control, I1 and I30. And as you can see, quite counterintuitively, the controls actually have the lowest survival and the unmated have the best survival. So as opposed to benefit of long mating, they actually suffer a cost of long mating. I'll come back to this in a little bit of time. Right. So now the third possibility is that perhaps the long mating has something to do with modulating the female remating behavior for the benefit of the males. And this is known in a variety of insect species. And to explore this, he conducted a series of double mating assays. Again, the design of the experiment is very simple. The first mating is with the treatment male, where the mating is actually interrupted at different time points. And the second mating is with a standard male. And of course, different components of remating was observed. So first thing is, no effect of the treatment on remating latency, intromission latency, but interestingly, there is a significant effect on remating duration. If you look at the plot there, I30 and I50, both of them have significantly higher remating duration. If you connect remating duration, if you use remating duration as a proxy for, say, post-copulatory success of the males, then to attain the optimal post-copulatory success, complete mating is, at least it appears to be necessary. Right. We still don't know why repeated intromission. We have some idea about why long mating, but why repeated intromission. One idea that we had was, let us look at what is going on during this inter-intromission phase and look at the behavior, perturb the behavior and see what happens. And that is where we looked at one particular behavior where males use their hind leg to rub on the abdomen of the female, and this is where we targeted our manipulation. What we did was to create 2 different treatment levels, one in which the male hind leg was ablated, and the other one was sham, so just any leg other than the hind pair of legs were ablated. And then the males were allowed to mate, and the mating behavior was observed. It turns out mating latency, mating intromission duration, mating duration, inter-intromission latency, none of these were affected, but there was something interesting about the number of kicks received by the males. And this is the other aspect. During this period, males rub the abdomen of the female, and the female invariably kick the males, almost trying to dislodge the male. And this is where what we found in our treatment males actually received significantly higher number of kicks, indicating that perhaps they are somehow sensorily stimulating the female to accept them and show less amount of resistance to continuation of the mating. So hence, leg rubbing seems to reduce female resistance. Inter-intromission period is probably used as a copulatory courtship. And what we are proposing is that the reason why they are showing this repeated intromission is simply that the males need to get out of the mating, get out of the copula, and perform this leg rubbing behavior so that the female can allow the mating to continue. Right. Well, I have been trained in sexual conflict. My strong interest is in sexual conflict. I see sexual conflict everywhere. And there is a very obvious scenario where the female is carrying the males around, so there must be some cost. 2 minutes. So that's what we looked at next. And just to cut a long story short, we looked at the effect of the length of the mating and number of mating, and looked at what it does to the female. Very briefly, what we find is that if you look at fecundity of the females, short repeated mating is actually best for the females. But in a natural population, what do? They go for long and repeated mating. Remember, in this particular species, females actually undergo several rounds of mating. That's first evidence of some sort of a conflict. Moving on. Yeah, in terms of survival as well, if you see the complete mating, multiple complete mating is the worst, so which is their standard condition. Any kind of interruption, any kind of modulation of their mating behavior, that actually is better for the females. The female which is allowed to undergo complete mating repeatedly, they die much faster, they have much higher mortality rate. So anyway, summing things up, what we find is that it's a rare mating behavior. Long mating modulates the female remating behavior. Of course, it has to be putative mate guarding strategy. You cannot rule that out. Leg rubbing possibly is a copulatory courtship that allows them to mate for a longer duration. Long mating seems to have a significant cost on the females. So, thank you. Thanks a lot for your patience. I'll end my talk by acknowledging all the lab members. Smita is going to present a poster that will be on Drosophila. I haven't left Drosophila. And ISER Berhampur, where I belong to, is an upcoming campus. All of you are invited to ISER Berhampur campus. It's a beautiful place. The campus is coming up. Hopefully in another 6 to 8 months, our campus will be fully developed. Thank you. Thanks a lot.
Any question, please?
Oh, we don't know that. We don't know. But it seems like males have some control over it, because the leg rubbing behavior seems to be important. So there is some control of the males. But we can't really specify that it is under complete male control or not under female control.
Yeah, Bodhi, nice talk. So are the females polyandrous? Do they store sperms from multiple males?
Yes.
They are. Okay.
Very standard insect mating system.
Right.
Females are very, very promiscuous. They have spermatheca. They have everything to store sperm.
So have you checked whether this long matings actually ensure sperm storage in the female?
Whether it ensures, no, we haven't checked that. But they seem to have a lot of sperm inside their genital tract all the time.
For insects who can store sperms from multiple males, sometimes the strategy to prolong the mating from the male's point of view is to actually ensure that his sperms stays in the system and gets a preference to fertilize.
Correct. One big problem with our system is that we don't have a mean to look at paternity.
Right.
That is going to be our next big project. We have to find a way to look at paternity success. But yeah, that's a very important question. Yeah.
And this kicking, does this kicking also happen to the control males, or it is only to the ablated males?
No, it is a standard observation. Control males as well, this kicking happens. When you ablate the hind leg, then number of kicks goes up, significantly goes up.
That is because female is perceiving this male as a defected male and doesn't want to.
Well, we don't want to put our interpretation to it. The point is, if the leg rubbing is not happening, which is going to happen if you ablate the hind leg, there is more number of kicks. Now, whether the female is considering the male as something or I don't want to put my interpretation into it.
Okay. Sir.
Following up on the previous question here, do you not see reverse sperm precedence in your beetles? In a lot of insects, there is reverse sperm precedence, so the last male to mate,
Yeah.
his sperm get used up
Yeah.
first. That's the first thing. That might completely change the dynamics
Correct.
of what she was talking
No,
about,
we
right? And the
completely
second question related
Yeah.
is that in most invertebrates, not just beetles, you often see a spermatophore, not just sperm, right? So spermatophore is a pretty large part of what males pass on in these
Right.
insects,
Right.
and that is just packed with nutrients for the female.
Yeah.
Right. So do you not see that in beetles in general, and
At least
specifically in this
here,
group?
at least here, we did not see any evidence as such. There is no obvious evidence. Our observation is, our interpretation is slightly indirect. We are inferring this from the observation that if you change the duration of the mating, it does not change the female starvation resistance. If they are receiving more nutrient or less nutrient, the starvation resistance should be different.
Hi.
You have observed longer duration of population in second mating.
Pardon?
Longer duration of population
Huh.
in second mating.
All matings. So yeah, so we do have some data on subsequent mating, but we haven't really got to a conclusion with that. But all the matings are pretty long. Yeah.
So what is significance of this?
Uh,
Second mating or repeat mating should have longer duration of population.
So that is what we tried to infer. What we think is that longer mating is required to ensure paternity, although we have to find a way of checking paternity. Once we have that, then we'll be able to provide more conclusive answer to this. But we think that this has something to do with ensuring paternity.
Thank you.
Thanks.
Hi. Yeah. Yeah. Hello. Yeah. Very nice talk. So I wanted to ask, like, with more longer mating, we expect that be more investment for the offspring. So lifespan should be our question. So why was it intuitive for you? You said that it was counterintuitive for you when you you did not expect longer lifespan.
Right. So yeah, so that the first survivorship plot that I showed, that was actually not longevity, that was starvation longevity. So that is starvation resistance. So our prediction was, if they are receiving more nutrient, then those who are mating longer, for longer duration, they should be able to survive better, right? They should have lower mortality rate, but they have higher mortality rate. That seems to be a cost of mating rather than benefit of mating, right? And in the last part, what I showed is that there is a clear evidence of sexual conflict, meaning while mating, the males do something which brings down female fitness. So we think that starvation resistance data is actually an outcome of sexual conflict rather than nutrient transfer.
Thank you.
Each intermission? It's a very difficult question to answer. Yeah.
Why I'm telling you, because in prime basis, well recorded, but
Huh.
SME system and MME system,
Right.
there's a single mate definite system.
Yeah.
Like the one in macaques, for example.
Right.
But line-tailed macaques are not MME system. It's a multiple mating to definite system. They have to mate several times actually to get to mate.
Right. So if you remember the sperm transfer plot, so one in intromission, then it goes up to 15, and then after 15 there is no difference, no change, right? So what we think is that it doesn't make any difference beyond 15. Exactly up to how many intromission sperm transfer keeps happening, that we don't know. But definitely there are at least certain number of intromissions which does not involve any sperm transfer. But even without sperm transfer, these intromissions can help in storage of sperm, utilization of sperm. That is an open possibility.
Hi, Bodhi. I just wanted to know whether you are planning to check the sperm counts at different intermittent matings whether they are being present in the, how much is present in the bursa storage organ, just to see the trajectory of the sperm during the prolonged mating?
No, we haven't done that. Our sperm count data is the entire sperm that has been transferred. So
Okay.
all,
Yeah.
yeah.
Okay, thank you.